Good morning, students. In the last lecture, we had talked about with the concept of the sexual reproduction, that is the structure of the androecium and the gynoecium, that is the stamen and the pistil. We shall now today discuss about the concept of the structure of or the internal structure of anther and how are the pollen grains produced. Now, if we look about the structure of the anther children, so we shall talk about its transverse section of anther. Now basically we have talked about the structure of the anther, that is can be monothecous or dithecous. We will be taking here the structure of dithecous anther children. Let us draw the anther children first of all, that is the dithecous. Now, this is the structure of the dithecous anther. There are 4 lobes, and in between here is the what children? That is the connective tissue. Now there are 4, we can say, microsporangium, we can say. That is this one. So these are the 4, we can say, microsporangium. Now there are 2 lobes. So this is the first lobe and this is the second lobe children. Now this is the connective tissue, 4 microsporangium, we can say. Now the space between over here, that is this one, that is known as the anther wall. It is known as what children? Anther wall. Now this anther wall has some layers in it. Okay. Now let us talk about it in one by one children. That is the first one, that is known as the epidermis. This epidermis is flatly arranged children. This one. So this is what children? Epidermis. Now basically this epidermis, so the first layer, that is the what children? Epidermis. The second layer is radially arranged, like in the form of this part. These radially arranged cells are known as the endothelium. That is, it is known as the what children? Endothelium. Now, the third one, that is known as the middle layer. Now, this middle layer is made up of, we can say, parenchymatous tissue. Or we can say it is made up of parenchyma tissue. This one, that is this part. They are loosely arranged in the form of 2 to 3 layers. This one. So these are the what children? That is the middle layer. Now, all these 3 we can say have the function of each and every one. Now, as the epidermis, that is having the function to protect the, and we can say the microsporangium. Now, apart from that, this endothelium, they have a substance known as the callose, or we can say calyx thickening. Now, the middle layer, they are loosely arranged and it is made up of parenchyma tissue. Now, parenchyma tissue has one function, that is to what children? That is to store the food, children. Okay? And the fourth one, which is the other one, that is known as the tapetum. Now, this is a very important layer, children. This tapetum is arranged inside, we can say this one, see like this, in the form of inverted triangles, as you can see, children. So these are the what, children? Tapetum. Now, if we talk about the characteristics of the tapetum, they are first of all multinucleated and they have thick cytoplasm. They have what, children? Thick cytoplasm. Now, there are two types of tapetum. We'll later on study this, children. Now, what are the functions of the tapetum, children? That is the function. That is, they are nutritive, or it acts as a nutritive layer, we can say. That is, it provides nourishment to the microsporophyle. Okay, another function is that they secrete enzyme. They secrete what? Enzyme called callose. Now, this is the enzyme which is being formed, or we can say known as the callose. Okay. Now, apart from that, this callose, we can say, which the enzyme which is being secreted, the callose, dissolves the, we can say, callose and help in rupturing or dissolving the pollen grains. Okay. Now, apart from that, secreting the enzyme, they also secrete the Ubish bodies. Now, these Ubish bodies are called sporopollenin. Now, this sporopollenin is the hardest material or the most resistive material known so far, children. Now, what it does, it protects the pollen grain from all the, we can say, external conditions like the water, humidity, temperature, other enzymes, other, we can say, external agents, we can say, so that it does not get harmed, we can say. Now, you can recall the example, or I can say, give you the example, like if you see the white powdery like substances, the yellow colored pollen grains which falls on your hand if you take the hibiscus flower. So in that we can say these pollen grains, okay, they are being having the yellow colored like powdery substance which is having the what class? Sporopollenin. Now this sporopollenin will make up the exine of pollen grain. Now what is the exine? That is the outermost layer, that is the exine and the intine. We'll discuss later on this part, we can say, children. Now this part, if you see over here, that is known as the point of dehiscence. Now this point of dehiscence is the point wherein the all the four layers we can say, there is epidermis, endothelium, middle layer and the tapetum. All these layers we can say, they will fuse during the maturation period or when the pollen grains are being produced. Now, but it is the point, that is this one, that is the point of dehiscence. This point of dehiscence is the point wherein the anther or the pollen grains will rupture, and it is only through this point the pollen grains will be matured and will be released we can say. From no other point this pollen grains has been or has to be released we can say, as because all these three layers they get fused or we can say they get tightly arranged we can say. Okay, so this is all about say we can say the structure of the transfer section of the anther. Now there is one other function of the tapetum, that is it also helps in developing pollen kit. Now pollen kit is nothing but the structure binding wherein the pollen grains they or the anther they mature themselves, or the microsporangium, that is this one, microsporangium. Okay, they start to develop and to get mature we can say, and they undergo the process of microsporogenesis we can say. Now microsporogenesis is the process where the microspores are being produced. Now this microsporangium has small spores inside them. They are known as the microspores. Now once the microsporangium matures, they develop into pollen sac, and this pollen sac they have the what children? Pollen grains we can say. Now these pollen grains which are being produced, they are being produced by from the mother pollen grains or the microscopic mother tetrad cells, that is the PMC children. It is known as the what children? PMC children. Okay, so I hope you are what you can say clear with the structure of the anther and the function of the tapetum. Now tapetum we can say, they are of we can say two types. Now there is no space so I am writing over here. The first one, that is the glandular tapetum, and the other one, that is the amoeboid tapetum. Now this glandular tapetum is secretory in the nature, whereas the amoeboid, it is known as the plasmodial in nature, as because as the name suggests, amoeboid, it having the secretory or we can say plasmodial nature. Okay, and this glandular having this secretory we can say substance or secretory in nature we can say. Now let us talk about the structure of the pollen grains we can say through microsporogenesis. Now microsporogenesis meaning what children? That is the process by which the four tetra microscopic mother cells are being produced. There it is known as the what children? The microsporogenesis. Now if we talk about the structure of the pollen grain, so the structure of pollen grain we can say, it is generally 20 to 25 we can say micrometers in diameter. Now generally it is made up of two layers, that is the exine and the intine. Okay, now we can say, so if we talk about the structure of this one, so this is the we can say the exine, and the other one, the inner layer, that is known as the what children? Intine, or the text one we can say. Now to this we can say exine we can say, they are having small we can say gaps in between. These gaps are known as the germ pores. They are known as the what children? Germ pores. These germ pores, they do not have the sporopollenin. That is, I told you, what is sporopollenin? A type of resistive material which is resistant to all kinds of different we can say elements while the anther is being protected, or the pollen grains are being protected. Now these germ pores, they do not have sporopollen. So wherein these are the small pores or the gaps from wherein the pollen grains, they will mature and they will be released with cell. Now the cytoplasm of the pollen grain is being surrounded by, we can say, that is the plasma membrane, we can say. So this is the what, children? The plasma membrane. Okay. Now inside the, we can say, internal, based on the cytoplasm, okay, or when the pollen grain is being matured, the matured pollen grain, we can say, contains two type of, we can say, layers in the intile, we can say intile. That is, let us see this one. So the first part, that is known as the vegetative cell. It is known as the what, children? Vegetative cell. And the other one, that is known as the generative cell. It is known as the what, children? Generative cell. Now, if we talk about, so it is spindle shaped, we can say, the vegetative part, and we can say are having 60 to 70% of the angiosperm are having two type of stages. That is the first part, that is the what class? The vegetative, and the other part, that is the what class? Generative cell, we can say. Okay. Now, it is being said that in many parts of the countries, we can say, in many parts of the countries, we can say, like the other western part of the countries, these pollen grains, they produce the, we can say, allergic diseases. Like we can say a type of, we can say, grass known as the carrot grass, that is commonly known as the parthenia. It is a type of crop wherein it causes the allergy because of the pollen grains. And to avoid this, these pollen grains are being, the pharmaceutical companies, they manufacture the pollen tablets. Now these pollen tablets are being given to the horses and the other plants and the animals so as to increase the efficiency, we can say. But it is some, what you can say, the legal in the western countries, whereas illegal in the other parts of the country as well. As we can say, we do not have the much more knowledge of this pollen tablets, we can say. So this is all about the structure of the transverse section of the anther and the pollen grain, we can say. I hope you have understood the topic, children. If not, then please do let me know so that we can clear your doubts, children. Okay. Now in the next video, we shall talk about the structure of the pistil and the transverse section and how do the pollination takes place. Thank you, children. Thank you very much.